Welcome friends in a e-learning section of 10th Science topic Heredity and Evolution. In this topic we learn about what is meaning of the heredity and how the process of evolution occurs simultaneously among the living organisms. First, we have to understand what is heredity. Heredity means the transfer of physical and mental characters from parents to their offsprings. नीट लक्षात घ्या. हेरिडिटी म्हणजे नेमकं काय? आपल्याकडे आपल्या आईवडिलांपासून जे आलेले कॅरेक्टर्स आहेत, मग हे कॅरेक्टर्स आपला रंग असेल, आपली उंची असेल, आपली शरीरयष्टी असेल, किंवा आपण ज्या पद्धतीने बिहेव करतो, ज्या पद्धतीने वागतो, हे सर्व प्रकारचे कॅरेक्टर्स असतील, फिजिकल अँड मेंटल कॅरेक्टर्स which come from our parents to us is called as heredity. Now the question in your mind may arise, how does heredity arise? Which are the some organelles which are related with this process? We discuss now. In earlier classes, we discuss about how the characters, how the cell is controlled, how cell work, how the functions of cell will be carried out. So we know about the characters. We know about the cell organelles of the cell. Among the cell organelles, there is one important cell organelle which is termed as a nucleus. We learn nucleus controls the activities of cell. Nucleus is important because nucleus is related with transfer of genetic material. Means nucleus is related with the heredity. So which component of the nucleus is related with the heredity? Because nucleus contains nucleoplasm, contains chromatin network, contains nucleolus, and also the nuclear membrane. So among these four parts, which part is related with the heredity? Here the component is called as the DNA or the chromosomes. During the cell division, chromatin network which is present inside the nucleus may start to separate from each other and the formation of the chromosomes may take place. This formation of the chromosomes may occur. Then on these chromosomes DNA are present. And so this chromosome or DNA are related for the transfer of the characters from parents to the offspring. So chromosomes are related with the heredity. We learned in the earlier class that how DNA is composed. DNA is the structure. DNA is the polynucleotide chain which is composed of the deoxyribonucleotides. So the single unit of the deoxyribonucleotide is made up from the ribose sugar, phosphate group, and nitrogen bases. there are the 4 nitrogen bases. A stands for the adenine, T stands for the thymine, C stands for the cytosine, and G stands for the guanine. So, among these 4 nitrogen bases, 1 nitrogen base is attached to the sugar and a phosphate group, and then they form the single unit of the deoxyribonucleotides. Large molecules of the deoxyribonucleotides, thousands of the deoxyribonucleotides may attach with each other and forms the polynucleotide chain, and this such long polynucleotide chain of the deoxyribonucleotides is called as the DNA. So DNA is related with the transfer of characters, particularly related with the heredity. And so, when we learn detail about the heredity, how this heredity is studied by the various scientists, discussed very short. The first scientist, which is known as the father of genetics, John Gregor Mendel, near about in 1866, carried out the large work, and from this large work he concluded 3 laws, and in 3 laws he found that in the cell there is some structure. We don't know which is the structure, whether chromosomes, whether cell organelles, whether nucleus, but for this he gives the name that the factor is responsible for such type of the characters which transfer from parents to their offsprings. Later on, in 1901, another scientist named as the Hugo de Rice, he worked on the mutation, that is the sudden changes occurs in the genetic makeup of the living organism. Why such sudden changes will occur are studied by the Hugo de Rice, and this Hugo de Rice is also the understood these characters, these changes may occur due to the mutations. Meanwhile, in 1902, another scientist, Walter and Stone, studied the paired chromosomes in the grasshopper of pasta. Any scientist before the 1902, they don't know that the chromosomes exist in a pair. Nowadays we can say homologous chromosomes, non-homologous chromosomes, but before this study, the scientists don't know the chromosomes exist in the pairs, and so this study is also important. And later on, different research proved that when the characters are transferred, these characters are transferred by genes, a small segment or fragment of DNA molecule which is responsible for such transfer of character. These characters are transferred from the genes. And after that, in 1944, 3 different scientists, Heredity, McCarthy and McLeod, these 3 scientists proved that all the living organisms, from bacteria up to the highly evolved living organisms, like human being or in plants we say angiosperms, DNA is the genetic material. But there are some living organisms like the viruses, which contains the only single type of the genetic material, neither DNA or neither RNA. So in such viruses, if DNA is present, DNA is the genetic material. If RNA is present, RNA is the genetic material. But except the viruses, all living organisms contain DNA as the genetic material. And then in 1961, scientists Francois Jacob and Jack monad learned and discovered that how the process of synthesis with the help of DNA in a bacterial cell may be carried out. So this is the important, that how the cell is related, how the DNA is related for the important function, that the protein synthesis. So important function in the cell is the synthesis of a protein. How this synthesis of protein is related with the heredity? This is another question we discussed later. Because the protein can formed then with the help of different techniques like the recombinant DNA technology. Scientists carried out to find how this DNA contains the different sequences or genes are related with the process of protein synthesis. So, here this is the important work from the John Greger Mendel, Hugo de Rice, Walter and Sutter, Avery McCarthy McLeod, Francis Jacob, and Jack Brown. All these scientists are related with this heredity. And carried out very important functions. They explain very vast scope of the recombinant DNA technology or heredity in the field of genetic engineering. Also, the science of heredity is related with the somewhat hereditary diseases, how these diseases may occur. So, what is the diagnosis? What is the treatment? And may this disease may be prevented from transferring from parents to their offspring. So, the heredity also helpful in the diagnosis, treatment and prevention of the hereditary disease. Also heredity related with the production of hybrid animals and hybrid plants. Hybrid varieties of the plants play a very important role in the field of agriculture, because for such increasing population, need of the cereal crops may always go on increasing, and therefore here we say this demand of the food is completed by the help of these hybrid varieties of the plants. So here heredity play a very important role, and also for the industrial processes where the microbes are used. For example, in the production of the vitamins and antibiotics, where the microbes are used. For example, we take the example of the industrial production of the insulin, which is related with these microbes. So, these all the aspects which are related with the heredity, that the chromosomes, DNA, and also the other type of the structures which are related with this genetics, are all focused on the process of protein synthesis. So, now here we discuss the process of protein synthesis. meaning of the protein synthesis? What are the uses of the proteins in our body? Protein synthesis, generally we know protein is the somewhat important component of our diet. So, when such proteins are we take from our diet, then what are the necessity to produce proteins again inside the body? Because the proteins which are taken up by our body through our diet are the somewhat raw material for the production of the proteins inside the cell. Because मग आता आपल्या मनामध्ये असा प्रश्न आला असेल की सेलच्या आतमध्ये प्रोटीन सिंथेसिस होणं का गरजेचं आहे? कशासाठी सेलच्या आतमध्ये प्रोटीन? आपण तर आपल्या आहारामध्ये प्रोटीन्स घेतोय, बरोबर आहे. आपण आपल्या आहारामध्ये घेतलेले प्रोटीन्स हे सेलच्या आतमध्ये तयार होणारे जे प्रोटीन्स आहेत त्यासाठी रॉ मटेरियल आहे. ज्यावेळेस आपण आपल्या शरीरामध्ये प्रोटीन्स घेतो त्यावेळेस अशा प्रकारचे जे प्रोटीन्स आहेत हे अशा प्रकारचे प्रोटीन्स आपल्याला माहित आहे ब्रेकडाऊन इनटू द व्हेरियस अमिनो ऍसिड्स. आणि मग हे जे अमिनो ऍसिड्स आहेत when transferred towards the cell, मग सेलच्या आतमध्ये आपला जो डीएनए आहे हा डीएनए वर कशा प्रकारचे प्रोटीन्स तयार झाले पाहिजे ज्याची आपल्या शरीराला गरज आहे त्याचा मेसेज तिथे आहे. आणि हा डीएनएचा जो मेसेज जाणार आहे त्याला आपण ट्रान्सक्रिप्शन असं म्हणणार आहोत. आणि हा मेसेज गेल्यानंतर जिथं मेसेज गेलेला आहे आपण 9वीच्या वर्गामध्ये शिकलेलो आहे जो मेसेज नेणारा सिंगल स्ट्रँडेड मॉलिक्यूल आहे त्याला आपण आरएनए असं म्हणतो. हा एमआरएनए मग न्यूक्लियसच्या बाहेर सायटोप्लाझम मध्ये येणार आहे आणि सायटोप्लाझम मध्ये आल्याच्या नंतर तो आपल्याला सांगता येईल की जो डीएनए कडून आलेला मेसेज आहे आणि जे सेल मध्ये आलेले अमिनो ऍसिड्स आहेत त्यांच्या साह्याने म्हणजे टीआरएनए हा आलेल्या अमिनो ऍसिड्सला त्या एमआरएनए आणि रायबोसोम कडे घेऊन येणार आहे आणि मग अमिनो ऍसिड्स तिथं एकत्र येणार आहे आणि मग प्रोटीन तयार होणार आहे. ही जी प्रोसेस आहे त्याला आपण ट्रान्सलेशन म्हणणार. आणि ही जी थोडक्यात थीम सांगितली डीएनए ट्रान्सक्रिप्शन एमआरएनए ट्रान्सलेशन प्रोटीन याला आपण सेंट्रल डोग्मा ऑफ द प्रोटीन सिंथेसिस असं म्हणतो. आणि म्हणून आपण ज्यावेळेस प्रोटीन सिंथेसिस शिकणार आहोत हे प्रोटीन सिंथेसिस शिकत असताना त्यामध्ये 3 स्टेप्स आहेत. एक ट्रान्सक्रिप्शन, दुसरी ट्रान्सलेशन आणि तिसरी ट्रान्सलोकेशन. याबद्दलची पण आपण माहिती घेणार आहोत ती नेक्स्ट सेशन मध्ये. ओके, थँक्यू.